Hello and welcome to our webinar. I'm very happy to have you here today. In our webinar, we want to address how CeGaT ExomeXtra solves patient cases that normally would have been unsolved. And therefore, we collected a couple of interesting cases that we have solved in the last 4 years. On that purpose, I do have Hans Gabriel here with me, who is one of our senior genetic diagnostic responsible persons and who will guide you through these individual patient cases.
And I'm here with Stefan Griesbach. Stefan is the head of sales at CeGaT, and he gives you an introduction of our company and how we work.
Okay, so thank you very much. In order to start here, what I want to start first off with is like who we are, what we are standing for. So CeGaT has been founded back in 2009, so we're looking at 15 years of experience here. So at that time, Saskia and Dirk Biskup founded the company. It was the idea of Saskia, our medical director, human geneticist specialized in genetic diagnostics, that had the idea and vision to use NGS technology in human genetic diagnostics that had not been done at that time. From then on, we substantially grown and have been running that business. We are a family-owned business. I'm very proud to say that. And what we are doing today is we offer these rare disease diagnostics. We offer raw data delivery services for researchers. We offer tumor diagnostics for patients. So we offer a broad spectrum of genetic diagnostics. And one of our key values is that we want to deliver something to our treating physicians, to our patients, to our caretakers that is meaningful and beneficial for the patient. We always have this as the main goal in mind. I don't want to go through all the details of this timeline here, just a couple of brief points. One of the points being, again, as mentioned, we are founded in 2009. We are looking at 15 years of genetic analysis. And one key aspect today is what you see here in 2019, the introduction of the CeGaT ExomeXtra. Up till that point, we utilized custom enrichment kits for diagnostic panels. And we've never been satisfied with using an exome enrichment kit because the enrichment kit that has been commercially available was not sufficient, sensitive enough to get the diagnostic yield we were aiming for. And 2009, we had been at the position to say we can design an exome that fulfills our needs. Of course, we are international operational and therefore we think it's mandatory to have accreditations. We are accredited according to the ISO 15189 norm. We are accredited to CAP CLIA norm and for the raw data services also to the 17225 ISO norm. And with all of that, we try just to do the best possible analysis that we can get to you. So when we're speaking about genetic diagnostics, and once again, you see Saskia here, and in the lower part of the picture, you actually see our brand new building that we are very proud to have moving in March this year. So when you look at genetic diagnostics, we have 3 steps in genetic diagnostics. The first one is that we sequence everything of relevance. The second one is that we use bioinformatics that helps us to streamline the process of diagnostics without getting rid of anything that we need. So we need to be carefully titrating what our filter criteria are and how we do that. And then at the end, very importantly, our medical reporting team. Because at the end of the day, there needs to be a medical report. And this medical report needs to reflect the current state of science and needs to be meaningful. We can do our job very easily if we give you just a variant list, but this is not the aim of a genetic diagnostics. The aim of a genetic diagnostic is to identify the cause of the genetic disease. And this is the goal to give back to you in the medical report. So in order to do that, we need to integrate all of these components, and this is what we're doing in-house. And today we're going to speak about exome diagnostics. So very briefly as a reminder, what is an exome? An exome is approximately 1 to 1.5% of the genetic code of a human. And it is defined as the sum of all protein encoding parts of this genome. So probably can argue about the detailed number, but we have about 23,000 genes in there. And when we look on the reported variants known, this is about 90% of the disease-causing variants that are present in public databases, as well as our in-house databases, that cause a disease are located within these exonic parts of the genome. And when we use that, we can do that different purpose, or we can do different services with that. The one thing is that we do a singleton exome, so we sequence the index patient, the patient that has a disease, the phenotype, and we analyze that, and this is what we call singleton ExomeXtra. Then we do a trio exome diagnostics, and that will also be the main topic today. We are strong believers in trio exome diagnostic. It just gives the best possible yield and most clinical insights on your patients. So I will come to that in a second. You can also do that in a prenatal setting. We actually have a very large cohort by now, over 2,000 cases on prenatal exome diagnostics, where we want to identify an ultrasound finding or screening purposes to identify something that is potentially not quite right with the fetus prior to birth, so that we can act upon that immediately after birth or explain a phenotypic symptoms that you can see in the ultrasound. So what is a trio? Trio exome diagnostic refers to a setup where we have an index patient and we have two parents. And of course, still we sequence all of those and we utilize the parents. They are not actively investigated, but we utilize these parents as a filter criteria to be able to determine on the index which of these variants the index has is a de novo variant. So a variant that has been not inherited from either of these parents. And when you look at genetic rare diseases, that obviously is something that could be very likely cause a genetic phenotype or a disease. Then we look for homozygous variant where both parents are heterozygous. So both are carriers coming together in autosomal recessive disorders, for example, and then we do have a phenotype, and we can also have something which is called compound heterozygous, when it's not the same variant, but we have two different variants in the same gene coming together and cause a phenotype. This is a very classical trio exome diagnostic approach, and Heinz will give you some details how that actually solves a patient case. Heinz.
Thank you, Stefan, for the great introduction on trio exome sequencing. This is our first case. All the cases I will present you within this webinar are real cases coming from our diagnostic department. So here are the original indication for testing. So we had a 3-year-old girl which was affected by developmental delay and motor delay, had macrocephaly and some other symptoms. So in such a case where you have not a real idea what is the underlying cause for the problems, we always recommend to do trio exome sequencing, and that's what we have done here in this case. And here you can see in one slide the summary. What you see on the left side, you see the sequence data of all 3 samples. In the upper part, it's the patient, and in the middle, the mother, and down you see the father. And what you can see here, I just want to go, I don't go into details with this. You see that here you have an insertion of a single base leading to a frameshift mutation, which we consider as pathogenic and which is not seen in the mother nor in the father. So it's a de novo event. And if you check this gene where we found this de novo variant, you see that such a variant leads to the KGB syndrome. And when you go in more details, you will see that the phenotype of this patient affected by the KGB syndrome are really similar to this patient. So we were able to solve this case by a classical trio exome sequencing. With this words, I go back to Stefan.
Yep, thank you, Heinz. So far, that's very nice and very great to have these cases, but exome diagnostic and exome diagnostic. So far, this has not been the most fancy case. As you see, a classical de novo variant, classical trio exome diagnostics. When we compare, or when somebody says exome diagnostic, it always seems very comparingly between all different kind of approaches. But it actually, and that might be a little bit stretched, but nevertheless, you can compare it to have this ultrasound picture from 1978, where, yes, this is a pregnancy, and this is a fetus in the middle. You can barely see that. And then we have an ultrasound picture of 2018, 40 years apart, and you see so much details, 3D animations and everything. So both is called ultrasound. But of course, when you look at it, the amount of details you get, the amount of how able you are to discriminate between something that is potentially pathogenic or not and see more details is dramatic. And actually this holds true also for exome diagnostics. How do I come to that? So I started off with you with this one here. So this is the classical definition of an exome, but there actually is a lot more that can be done. And When we looked at that, and this is where we have been 2009, since, okay, what do we need to do? How can we get everything of clinical relevance? And coming back to this one, again, we have to work on all 3 steps. The first one is, let's talk about what we sequence. And when we looked at it and said, okay, we need to sequence more than the sum of all exons. So what we actually said, it's not sequencing the relevant genes, it's sequence all relevant targets. So where we ended up is, we said, okay, and this is the current version of our CeGaT ExomeXtra. It starts, of course, with all protein encoding genes. But then we do have clinically relevant RNA genes, something that is missed in a classical exome. We do have about 38,000 intronic intergenic variants, again, something that is not part of a classical exome. We do have the mitochondrial genome, per definition, also not part of the exomic region. We do have pharmacogenic relevant variants specifically enriched, and in our newest update, we also have included a backbone for detection of copy number variants throughout the whole genome in an RCGA-like approach. And this gives so much more insights that helps to solve patient cases that would have not been solved with a classical trio exome. And with this one, I would give once back to Heinz, who has now a handful of cases for you that explain how that translates into medical reports not been solved before.
Okay, thank you again, Stefan. So the next cases I have selected are really showing the difference between off-the-shelf exome and our ExomeXtra at CeGaT. So the first case, and as Stefan already mentioned, we do a lot of prenatal testing. So this is a prenatal case at a 27 plus 5 gestation week, and we have a skeletal phenotype. And if you are working in genetics, you know, if you have in a prenatal setting a skeletal phenotype, you have a high chance that this is caused by a genetic variant. Previous test was a microarray which was come negative, and we were asked to do a trio exome sequencing. And what we found here were 2 variants. You see in the upper part at the fetus, one was coming from the mother, the other come from the father. And this gene, the RMRP gene, is a non-protein coding RNA gene. It is known that this can cause metaphyseal dysplasia, which is a skeletal phenotype. So we considered this case as solved. And if you look for the coverage of this gene in a commercial kit, you see there's almost no coverage in a commercial kit. So because RNA genes are not enriched in a regular exome kit, and so they had missed this variance, and so therefore in a regular trio exome approach with a commercial kit, there had been a negative testing result. We could solve the case, and what is this gene doing? To give you some more information, as I mentioned, it is an RNA gene which is not protein coding, and it forms this hair loop pin structure and is a regulator of transcription of some upstream genes. So it is a good example how we can solve, even in a prenatal setting, cases with our ExomeXtra, which would not be possible with a regular kit. So therefore, I will show you some more examples. And I really love this case because it shows you a situation we are facing quite often. So here in another lab, already exome diagnostic were performed. And what they found was a pathogenic variant in the PLA2G6 gene. And in a boy aged 10 years with hypoplasia of the thymus and cerebellum, epilepsy, it was severe affected, had healthy parents. They found a pathogenic variant. So what is this variant doing? It's doing so it's causative for neurological disorders. And if you go into details, you see that the symptoms of the patients are really consistent with PLA2G6 associated disorders. However, the problem here is that there was only one pathogenic variant detected, and we have an autosomal recessive trait. Therefore, simply one pathogenic variant is missing. And that's a situation we see quite often in diagnostics. And so the lab asked us to do a test for this gene. And here are the results. First of all, we were able to confirm the finding of the other lab, so we could also detect the pathogenic variant, which is well known and well described. But we also found a second variant, and as you can also, if you are working in genetics, you will see from the nomenclature that is some special variant, which is a deep intronic variant. And we detect this variant as it were recently described by Boyer et al. As a causative mutation. And if we compare our enrichment with this of a commercial exome kit, you see there is no coverage in a commercial exome for this deep intronic variant. We have this enrichment of known pathogenic intronic variants, and therefore we were able to detect the second variant, and therefore we could really make the diagnosis of a PLA2G6 associated disorder. Another example, Stefan already mentioned mitochondriopathies, and here we have a 30-year-old female. This is just a recent case we just diagnosed a couple of weeks ago. He has a rather complex disorder. There was a kidney disease. Also, there was a short stature, diabetes, optical atrophy, hearing loss. So you see is really affected. Many tissues, many organs are affected. And there were already a mitochondriopathy suspected before. So they did a karyotyping, a muscle biopsy, and did genetic testing for MELAS and NARP, which are mitochondriopathies. And so we do a single ExomeXtra approach. And what you can see here, these are really variants from the mitochondrial DNA. We found a variant in the mitochondrial DNA. And if you compare it with a commercial exome kit, there is really almost no coverage. 23, normally you cannot do diagnostic with such a low coverage. We have a coverage for more than 200 at this region. And what is the result? Maybe you will wonder that there are two genes annotated. The point is, this is a special situation on the mitochondrial DNA, that both genes are sharing the same regions. They're using the same reading frame in some instance. So there's one variant affecting two mitochondrial genes. And what you also can see here is that we have only positive reads of 22%. So if you're working with mitochondria, you know that we have the problems of heteroplasmic variants. So it's not like in a regular setting where we heterozygous or homozygous variants. It can be everything between 1% to 100%, what we call heteroplasmic, meaning that that only a part of the mitochondria are really affected by the mutation. And if we look for the resulting phenotype, we found it's still unclear variant of unclear significance. We are so not absolutely sure whether this is causative for the phenotype, but there's a high chance that this is really the cause. And as two different genes are affected by the same mutation, we have also a slightly different classification. That's because the ATP8 gene, the clinical relevance of the ATP8 gene is not that clear. Therefore, the classification is a little bit more unclear than for the variant in regard to the ATP6 gene. So what the point is, to solve such a case, you need a good calling algorithm for mosaicism, for heteroplasmatic variants, and also good coverage for mitochondrial genes. The next case, it's a 1.5-year-old female, so a little girl, and again, not specific phenotype. We have global developmental delay, especially speech and motor delay, microcephaly, no obvious dysmorphic signs, and again, healthy parents. And with this next examples, I will show you something about CNV detection. It is so important to have a good algorithm which is able to detect deletion duplication within the whole genome. And what you can see here is the blue line means there's a normal copy number, and we zoomed in a special chromosomal region, and what you see in the patient is that at some point we have a lower coverage than the average, so meaning that's are the yellow line, meaning we have a deletion which is not seen in the father or in the mother, so it's a de novo event. And when we look at this in more details, we see that it's a 288 kb microdeletion at the chromosome and of the short arm of chromosome 17, and then you check the database, and these are well-known microdeletions which are associated with low mental delay and mental retardation. So we can solve this case. So I know that a lot of people still doing microarrays before they go to the exome sequencing. So the question is it still necessary to do microarrays? Or to rephrase the question, is this really microarray-like, this approach? So I will go back to Stefan, who gives you more details about our CNV backbone.
Yeah. Thank you, Hans, for these interesting cases. So, as Hans mentioned, we have been doing deletion duplications for many years. But, of course, when you look at it, how we're actually doing that is, so we do have a coverage plot. So we look at the probes you see here on gene A and GB, and we do look at the coverage expectations. If that is reduced to the coverage expectations, you call this a heterozygous deletion. Of course, homozygous deletions are quite easy to detect because there's simply no coverage at all. Nevertheless, there's a certain error rate and everything, but it's very good working, and we can go down here to single exon deletions, and we can find 1, 2, 3 exon deletions, 1 gene deletions very nicely. But what is about the part in the middle? What's about the part between these genes? And when you do an array CGH, what is one of the benefits of these array CGH? And so when you do that, what we find is that an array CGH, how it's normally run, has about a 50 kb resolution everywhere in the genome. And so we have been thinking, can we cut short on the diagnostic purpose, especially for cases where you do an array CGH first and then say it's negative, then we expand to an exome. Technology has been evolving. Can we cut that short? Can we integrate this in one test and just do an exome that is covering that? And so what we did is we designed backbone probes. So probes that in the intergenic space have approximately 50 kb apart and that are used solely for the purpose of detection of genome-wide copy number variants. And what we do with that is that we say we have a high resolution encoding areas down to single exons, but in the intergenic space we have a 50 kb resolution comparable to a 180 K probe microarray, how the standard is used in genetic diagnostics. And so this adds genome-wide CNVs to the CeGaT ExomeXtra, and thereby you can cut short on doing a stepwise approach with a microarray followed by CeGaT ExomeXtra. You can just do it in one test and get the same kind of results. And how that displays in real-life scenarios, Heinz.
Thank you, Stefan. So how it looks like. So here you see our old exome kit without a backbone, and you see that in this intragenic region we have no probes. So a CNV, a deletion duplication, is not possible to detect. And down you see the coverage, so probes, you see our 50 kb spacing. And is it of relevance? And if you look in this region, there's a reason why I have chosen it. You see there are no probes in the old version, while you have these nice probes in the new version. The relevance is shown here. So it is known that in some regions of the genome, deletion duplication can cause a phenotype, even when there is no protein coding gene included in this region. Here's an example which was also presented during the quality assessment scheme of CAPCLIA last year, where they have an example where a deletion in this region can be causative to disorder. This is well known for this region because in this region we have regulatory elements which control the downstream SUX9 gene. And if you have deletion or duplication in this region, it can result in a sex reversal phenotype, and therefore it can be important to have such deletion duplication detection outside the exomic region. And therefore, this is easy to detect such a disorder with microarrays, but with a regular exome kit, it is not possible, and normally you don't have any kind of probes within this region. So this is a good example where it shows you the clinical relevance of such an CNV backbone. So we think that microarrays are not necessary anymore if you run our ExomeXtra kit.
Thank you, Heinz. Yeah, so what we wanted to show you with this part of the webinar is that the sequencing aspect, the wet lab aspect, designing an exome enrichment that covers everything you want to have in a clinical setup is very, very important. Nevertheless, the subsequent steps are at least similar important. Already, Heinz mentioned that you need to have a certain bioinformatic filter criteria to be able to detect heteroplasmic phenotypes that are down to very low percentages, that this is very important. And when we look at the bioinformatic analysis pipeline that we developed over the last couple of years, of course, we need to integrate more and more data there as well. And what I want to show you here today is as well, there are different versions on that one. So we do have something what we call the iVerb filter. So in our trio exome pipeline, when you have both parents and you have an index, there's one blind spot. That is, if one parent is not affected, but still transmits the causative variant singularly to the child. And this can happen in different aspects. This can happen because we have an imprinting gene that is only affecting if it's inherited from the mother, for example, but the mother has inherited from her father, this father from his father, and that was a de novo event or something like that. So even there was nothing in the family, this would be inherited. In a classical trio filtering approach, this variant would be sorted out and you would miss it. So that is very important. The other one is UPD, Uniparental disomies, something that is also very, very interesting and relevant, and we have cases for that. We do have already shown you that the array CGH-like copy number variant analysis is very important. Of course, the bioinformatics, doing that, calling that, is extremely important here as well. We do have single nucleotide variant with copy number variant combinations. We do have repeat screening. So there's a lot of things that you can use in day-to-day work to enhance the wet lab data in a way that you solve more patient cases. And with this, we want to go a little bit, or for these, we want to go a little bit more in detail. So Heinz, again, with more cases.
Thank you, Stefan. Imprinting disorders are really important in diagnostic. Here we have a patient with overgrowth, macrocephaly, macroglosia, and also kidney disease. And here what you see the data from our trio exome sequencing. And you can see a variant in the gene CDKN1C in the index patient, which is a pathogenic variant coming from the mother. The mother is not affected. However, due to the imprinting defect, the maternal allele are imprinted and therefore not active. And therefore, in the index patient, we have a pathogenic variant which is active and therefore resulting in the Beckwith-Wiedemann syndrome. Another example will show you how important detection of mosaic variants is in diagnostic. We have a segmental overgrowth, a hemihypertrophy, as you can really see from the picture of the patients. We have also a developmental delay and motor deficiency. And we found a variant, pathogenic variant in the PIK3CA gene, which results in a neurodevelopmental disorder. And as you can see here, only in 22% of the reads, we found the pathogenic variant. And here we have a tissue-specific mosaicism, and which results in these also tissue-specific effect that only one side of the patient is affected. And we have our pipeline is able to detect pathogenic variants in mosaicism larger than 5%, in some cases even lower. And with these words, Stefan will tell you something about uniparental disomies.
So, thank you very much, Heinz. Before I start with the UPDs, maybe one additional word to the mosaic phenotype, and that is that coverage is very important here. Because if we want to detect something with allele frequencies of 22% or going down to 5%, we need substantial coverage on that position. So in order to detect a 5% mosaic phenotype, we need 100x coverage in order to discriminate between PCR artifacts and true mosaic variants. So coverage is a very important point, something, and that might be an argument you could make when you look at us and say we're targeting a lot of intronic, intergenic and RNA genes, is why don't you do a genome? And this is one of the exact reasons for that. And genome does simply not have the sensitivity, and especially not if you go down to mosaic phenotypes. That's impossible to detect in a genome that is regularly done with 30, maybe 35x coverage. So, but we wanted to talk about UPDs here. So UPDs is something that is a little bit special in that sense, because it does not occur all the time. With the UPD, what it actually means is that we have parental disomies. So we do have still a situation where we have two alleles, but the two alleles are not inherited one from mother, one from father, how it is for the majority regularly done, but it can be that it's a partial of a chromosome or a whole chromosome inherited only with both alleles from one parental side. And you can have a lot of different setups here. You see them depicted on the screen, on the slide. You can have maternal parental disomies, you have paternal parental disomies. They can have the same allele in two copies being transmitted into the child. It can be that both copies of one parent are handed over to the child and the other copy of the other parent is not inherited. So there's a lot of different options here that can be done or that can happen. And these most of the times are rescue mechanisms of the cell that at some point had a trisomy or partial trisomy at that point, and there was a rescue mechanism for that cell to survive, and it got rid, and let's say, on the wrong version. And that can inherit a lot of different problems, for example, due to imprinting. And so what we looked into that is said, okay, we need to be able to detect those as well in order to solve cases, because they are very, very important. So we tweaked our bioinformatic pipeline. I think there was a lot of discussions. Anyway, at the end of the day, we're now able, since quite some time, to detect isodisomies, heterodisomies, both paternal and maternal, of course, and they're standard-wise included into our exome diagnostics because of the unique setup when we are looking at heterodisomies, so where there are two different alleles inherited for one parental side. It only can be done in a trio exome because otherwise it would be hard to discriminate on bioinformatics. When you have isodisomy, what you normally look for is stretches for homozygous variants that would not be explainable in a different phenotype. If you have both parental information, you of course can also identify that these stretches of variants are inherited from one side. And this is something that we include in our trio exome diagnostics, and we report, of course, if they are phenotypically relevant. And how that looks like in a patient case, once again, Heinz.
Thank you, Stefan. So the example I show you is the case of a 3-year-old girl which has delayed motor and speech development, hypertonia, short stature, on constipation and obesity. And we already performed a single exome sequencing in 2021. But the case was not solved, and therefore we performed 2 exome sequencing 2 years later. And these are the results. And as you can see, we were able with our new algorithm to detect a hetero-UPD of 15, and we could show from the variants that it was a maternal UPD, and which also includes the Prader-Willi-Angelman syndrome. So we were able to make the diagnosis of Prader-Willi syndrome. And if you remember the phenotype is really consistent with the Prader-Willi syndrome. So we were able to solve this case. And this example shows you how important it is to have an algorithm which is able to detect such hetero-UPDs out from the trio exome data. And Stefan?
Thank you, Heinz. So with these, actually that was, I think, the last sample case we have for you today. But nevertheless, there's a next step in the diagnostic process as well. Of course, this has all been already, and you saw some glimpses of that during the process right now, but the medical interpretational part. And for the medical interpretational part, we had been thinking what we can do last year actually to make our medical report better usable in clinical practice. And what we came up with that we rebranded, or not rebranded, but we restructured our medical report and gave more clear-cut information in that could be utilized more easily for you. And I will walk you shortly through the medical report layout, what we have here. So this is how our germline report looks like. And what you see is on the upper side, there is a short patient summary information. You of course get what the order was, what the indication was. If that is a report with significant findings, you do have the primary results, so the gene, the variants that have been identified in that case that contribute to solving the patient's phenotype. In that particular case here, it's a pathogenic variant. We do have a conclusion, we have a clarification of the reported variant, and the recommendations on the genetic relevance. So all on the page 1 is the condensed information that you most of the times need. But if you want to dig in a little bit more deep and get the details, on the second page we provide that details. And what it gives you here is one example how that looks like. And we have under each variant that has been reported, each gene that has been reported, a statement about the phenotypic overview. What does this gene actually does? What is this associated with? Then there is a text which explains the gene function, that explains how the associated phenotypes are there, and there's of course references in there. And then you have the lower part, which is relatively new, which is a variant classification in a tabular flu. And that looks a little bit more, let's look into that one a little bit more in depth. So what this one has here is it classifies, and of course we do classify our variants according to the ACMG, ACGS criteria. It gives you a detailed rationale which criteria have been used for that particular variant for classifications. So you see them on the very left column. Then you have a point value that has been associated with this criteria and the description of that. And in case needed, there's also the citations on the publications that we base this classification on that has been combined to that. And then at the very bottom, what you see there is the point value, the overall point value that specific variant got. In that particular case, it was 11 points, which is according to the criteria a clear pathogenic variant, which is at the threshold of 10. But you also see when you look at the WUS scale that this is very fine defined between the individual criteria. So you have a tepid WUS, you have a warm WUS, a hot WUS, and so on. And you, as the physician, can understand exactly, when you look at the report, how we come to that conclusion. And of course, we are very happy to take questions at any point in that setup. So when you have a report of ours and say, okay, I will have a double check on the specific criteria, can you give me more details on that? Very welcome to do so, and we will explain to you the rationale. And we'll of course hear if there's any, if you have any opinion on that. So very happy on this exchange. So this is something which had very positive feedback to change this from a free text to this tabular view that gives you details on that. Another thing I want to point out is that we strongly believe in collaboration. I think we as the genetic field need to work together to enhance. There's so many still unknown things out there, so many things that needs to be there. So what we do is we upload our fully criteria-driven variant classifications up to ClinVar. So right now we're closing in on 100,000 submissions on ClinVar, and thereby we are the 6th largest submission entity to ClinVar. And we are very happy again to collaborate with everybody and to exchange information on the particular cases. One small word to our US customers. Some of you might have seen the news that we have been expanding back to the US recently. And here I want to just give you a very brief overview that we are accepting right now all major insurances. We have our in-network partnership is growing constantly. We have a billing team on site so that every patient is individually addressed, and the team will work with the patients for the benefit analysis prior to the starting of the testing, so the patient has the full control. We will never ask for money, so the patient at the time point of decision knows what they're ending in, and that's it. And they will understand exactly their out-of-pocket payment. And we have a US team that is helpful on that one. Very happy to take your questions there. Please visit CeGaT.us, and there you will have our 2 colleagues who will take your questions on those. So with this, I would like to conclude our webinar. Thank you very much for your attention. We're looking forward to answering your questions, and if we cannot answer your questions right away, or you come back later with questions to us, you do have an email address here. You can also find myself, you can find Heinz on our homepage. We have an email address that is very easy to remember, firstname.lastname@cegat.de. Just send us an email and we're very happy to answer them, even if you cannot do that right now. So, thank you very much.